Good morning, students. Today we will discuss about chemical storage and waste management. These two contents are really very important with respect to the lab safety course. And we will thoroughly discuss about, although like we will not go in detail about like some chemical properties, etc. But we will cover like the basic things about the chemical storage and waste management. Since we already cover about the basic safety guidelines or lab safety guidelines in the beginning lectures, but of course off and on according to the contents of the lecture. So maybe I will remind you about the certain or special or specific guidelines about the lab safety. So for this lecture, of course, there are certain guidelines. For instance, it is the responsibility of each person. So any worker, whoever enter into the lab, so they should understand, they read the instructions on the main entrance door. And of course, that's really important. So he also understand or remember about the hazards those are associated with the potential hazardous materials as well as the equipments. So of course, the laboratory equipments also have some hazards, so that we will cover in the equipment safety lectures. But of course, this all information is really very important if someone is going to enter into the lab for working purpose. It is also the individual responsibility to follow to practice the following general guidelines. Okay. So in couple of slides, you can see or you can find like the few guidelines, especially I prepare for this lecture. For instance, whenever you had a chance to enter into the store or the storage area where the chemicals are stored, then definitely you need to wear the eye protection PPE. Although like the basic PPE you already wear, for example, the lab coat as well as the gloves. But this is really important because we are not sure like which type of chemicals or maybe evaporate in the storage area. There are because different chemicals are stored there, and there are chances like sometime the in the air, or maybe like some the chemicals can evaporate into that particular area, so it will be like kind of condensed environment. So definitely, so you have to wear IPP especially in that area. And this is really very important. Don't use contact lenses, because for instance, once you visited a storage area and after that you can wash your eyes if you are needed, and sometime if you wear the lenses and might be there if they contaminated, so it will stay in your eyes. And I don't know like you have like good care or not, like. Okay, so there are chances like they can cause like kind of irritation into your eyes. Okay, so this is a kind of one guideline. I hope you can follow this one. Secondly, this is really very important, the knowledge about the hazards. Those are associated with certain chemicals that you are going to use. So as I mentioned in previous lectures, whenever you are going to use any chemicals, you definitely have like a full information about that chemical. And definitely, of course, the hazard is one of that information. Next is always, of course, wash hands even you just visit the lab or the lab area, whether you did your experiment or not. Okay, so always wash hands, and of course before leaving the working areas. And next is so this practice is very important, and more practically, don't never forget about this actor. So if you are working on certain chemicals, whether this is a different salt, acids, or radioactive material, or any other solution, or any other bottle, or any other stock, like whatever, so please don't remove that particular chemical into that position. If you are going to use it, so put back into that original position, because everybody know like where those chemical are specified into that particular area. So if you remove or move the position, maybe not the next person who wants to looking for that certain chemical, so maybe they will not find out. Okay, so this is really very important with respect to the storage. The next one is use equipment and hazard materials only for their intended purpose. Okay, so if your purpose is like like which experiment and which chemical you need it, so particularly for your whatever your purpose or the experiment, use any equipment or like any material. For example, if I talk about like the equipment, so in our labs we have ice making machine, so that ice making machine only will be used for to make ice for the experiment. Okay, so not like you make the ice for like for instance your like the drinks, right? So this is one example of equipment. There are other certain equipments that can be used like for example personal or domestic uses, but we will not use because those are specified for the labs. And similarly, the hazardous material, for example, there is a sodium chloride salt in the lab, so you cannot use that as sodium salt. Okay, and there are many other, the big list about the chemicals that can, for example, ethanol. So most sometime might be like some people they wants to use outside the lab. Okay, so there are certain chemicals, although they have some properties, they can be used for some domestic purpose, but of course now they are specified. Okay, so now they are specified for the lab and don't use for domestic or other purposes except like only for the intended purposes or like your experiments. Never leave an experiment unattended while it is like, especially for some equipments, if you are working those are producing heat or flame, as well as some chemicals if you are working and they are producing like reactions, and because those are like depend or the based on the limited timing, and if you are away from that particular experiment at that time, so it may cause any explosion or some dangerous or hazards for other lab fellows or in the lab. Do not use cracked, damaged or chipped glasswares. So this is very important for your safety. So if you are going to use any glasswares and you see like this is a cracked or damaged, so don't use it and just dispose of into the particular area and use the new one. Laboratory water sources and deionized water. So there are in commonly we use very good water like double distilled water or deionized water for the polymerase chain reaction like the PCR. Okay, so that's really very pure water, but of course again you cannot use for drinking or other domestic purposes. Okay, because again this is not for the health use, so this is for the laboratory purposes or for the laboratory use. So lab materials should never be consumed or tasted. So this is in general like a terminology, although this contain like some hidden information, because those chemicals or the materials that belongs to lab, of course they are not 100% sure like those are can be used for the eating purposes or some like these domestic uses. Okay, so fundamentals of storage. So how we can store different chemicals, the information from where we can get the information. So as I mentioned in the last lecture, MSDS. So this is the document that give like a whole information about any chemical. So whenever you have the list of this, the document of particular chemical, and there is a mention about the incompatibility, compatibility and different temperatures and the storage area like these things. Okay, so you just have to identify the chemical according to the MSDS information and then decide the location. And secondly, isolate and separate incompatible materials. Of course this is really very important. So the compatible materials, of course they can be stored like next to other. Okay, so that's no problem, but incompatible cannot. So in last lecture I gave one example. So sometime like incompatible materials, they can react with each other and can explode. So how we can sort out those like with particular area or the aroma? So it depends on the quantities of the chemical and the properties of the chemical and packaging. So these three properties are very important that give us a degree of isolation dependence. Okay, so for example, if there is a small quantities, might be it is the store like next to the incompatible material by chance I'm saying. Okay, but of course if like it's the big quantities, and of course this will be dangerous. In this slide is mentioned very beautiful for 5 different areas for the storage, and you can see here the first green, this is the area 1, and here you can see it says like low hazard area. So this is a low hazard area, and it means the chemical those have low hazards, and you can found those chemical from MSDS document, and you can store into that particular area. And then the flammables excuse me, so the flammables chemicals or liquids you can store into the area number 2. So this is showing like a the different red color, and corrosive chemicals of course you can group into the third areas, and of course they have like the white codes. Oxidizers always comes like a yellow colors, and of course so these are fall under like area 4, and you can store them like in the yellow labeling or like cabinets like these things. And then of course like the blue fall into the area number 5, like poisonous materials. So in this way, like according to the nature of the chemicals, we can divide it or distribute the chemicals into particular areas according to their properties. And of course off and on we remind like each facility the lab is furnished or equipped with these facilities: eye washer, safety shower, emergency phone, fire extinguisher numbers, spill material kits. So these always off and on I will remind, or I add these information, because this is really very important if you have like that particular storage area in the lab. In this slide you can see a best the ideal setup for the storage of different chemicals. You can see like for example the acids are stored in this direction, and then next to acid is a bases, and then dry chemicals, and then flammable cabinets, and then metal salts, okay, and then the spill materials, oxidizers, and like some other materials. So this is the look, the best or like one kind of ideal setup. Okay, again it depends like according to the store, the construction of the stores, or the location where somebody has like so it is not like this is kind of one example. Okay, but someone according to the their facility, so they can also change, or they can of course they can find like a different setups. So again, like of course this information is basic information is there. So here you can see the yellow color flammables, the cabinets. So in most of the labs, or I think in the biology labs, in each lab you can see like these are the these kind of storage area. So this is so these cabinets are for the flammable chemicals. And of course it must be labeled with this sign like these are the flammables. So of course nobody can keep like a fire, or cannot bring the fire close to this shelves. And of course their locks is not like a very simple locks. So this is a kind of these shelves are very specified to store these flammable chemicals, and they actually locked from the top, and then from the bottom, and then of course from the sides. So these are very important locks because this is a very sensitive chemicals. Okay, so 3 types of top, bottom, and here the sides are always locked. And secondly, you can see practically if in future you had a chance to visit any lab, bio labs, you can see these yellow cabinets are not like very big, so they are very small, because these are flammable. The shelves are like the cabinets, and always it does not allow to store like a huge number of chemicals. So there it has some certain capacity or limits. So for instance, not more than 60 gallon of class 1 or class 2 liquids are stored into this the cabinets, and not more than 120 gallon of class 3 liquids are permitted to store into these cabinets. So this is the maximum capacity. So you can see like that's why the size is always very normal. As we discussed in the previous lecture, there are certain like code numbers are used in the lab, and similarly here you can see in the labs commonly we use different acids and base storage chemicals. Always like when once like the different chemicals come, and of course they also use these different like the cap codes. So you can see you can experience or you can observe these color caps in the labs very commonly. So for instance, this red is used for nitric acid, and this blue is used for hydrochloric acids, and this the red is used for nitric acid, sorry, yellow sulfuric acid, and then of course brown is used for acetic acid, and white is used for phosphoric acid, and green is ammonium hydroxide. So if you you remember this information, and then if you are working in the lab, so just according to the cap color, so you can recognize so which one is that particular acid, although there is also labeling on the on the flask. So there is also labeling, but of course you will not mix these caps with the, or you cannot switch these caps with the with each other. And then there are certain colors, for example clear, black, orange, purple. So these colors, so this is for the stock. So stock mean like concentrated chemicals. Okay, so these colors codes are used, caps are used for the concentrated or like the stocks. But of course sometime you make like dilutions, for example 1%, 2%, 10 millimolar, like these. So for dilution, so you can use these colors. Okay, so different these colors, they are not specified for particular acids or any chemical, but these are the different color you can choose, and but these are only for dilutions. Okay, here you can see very beautiful video. I would suggest you just go on to this link and use you watch this video. So this is showing like different explosions of different alkali metals, or like once they add into the water, and how you can observe like how the explosion is happen. So here is the examples of some alkali metals with the reaction with water. Okay, so just to watch this video, this will be really helpful. So of course, as I mentioned before, like how we can store these chemicals into the storage area, or particularly like into the store or in the cabinets. But what are the chemical storage that does not need? We should not do these things. For example, don't mix waste chemicals in the same storage container. So like you had experience, or maybe like in the high school, so while you are working in some chemicals, if you use one acid, so even if it's used, but it's look like the original one that you can use, like okay, so not like mix again back to the original container. So once you use, then it will become your the garbage. Okay, so this is a waste. Okay, not like you can put back into the same container. So mixing two or more waste chemicals in one container can cause quite the violent reactions. So for instance, if you used different chemicals, of course don't mix like together into the same storage. So there are certain then again, of course, according to the wastage, so there are certain like the storage the baskets kind of the plastic you can see like in each lab, so they are labeled with different chemicals, and you can just follow the instructions. So don't store chemicals near heat, sunlight, of course other substances like okay. So of course the main idea is of course they can react because as I mentioned in the previous lectures, so each chemical has certain flash point temperature. Okay, and they can react with if we can store like, or if we would store close to heat or sunlight. Okay, don't store chemicals on floors or other places. So only store the chemicals into the specified areas, in the storage, into the yellow cabinets, into the poisonous places, whatever you, okay. So each lab has the specified area. So you can, once you start working into that particular lab, of course, this is the first thing you check, like it take like 1 week to understand the lab, about like the setting, about the storage, about the working benches, about the equipments, like these things, okay. So don't add water in concentrated acids. As I mentioned in the previous slide, there is one video about the alkalis. So there is a reaction. So similarly, so we should not add water into the acids, okay? And because of course it can also cause reaction. So don't store ice, dry ice. Of course, it has cryogenic properties in a sealed container. Of course, then of course it can cause also explosion. Let's move on waste management. Uh, so what is actually the waste? So waste is any substance which is discarded after primary use. So in lab, while you are working on certain chemicals or like liquids, dry chemicals, etc. So you can see even after uses it's look like sometime original, like in the original form. So even then you cannot consider it's original. So this is a waste, okay. So once you have used any chemicals after primary use, so it become worthless. So don't deal it as an original, don't put back into the original stock bottle, okay. So because you already use it, so it already act, it's okay. So it's already in the active form, it's already perform its function, and you then after using you can just think about that particular chemical is a waste. And of course a hazardous, if we talk about like hazardous waste, so the waste that has property can make like a dangerous or potential harmful effect to the health or like the environment, so those are called hazardous waste. There are, it could be like a solid, liquid or gas, okay. It could be so any form, so whether it's liquid, gas or solid, okay, that can cause any hazard or potential hazard to the human health or the environment. And of course the shops, so these are also the big source of shop waste into the hospital as well as into the clinics and in the college of medicine especially. And also of course in other biolabs where we are using different syringes and objects like those have like edges or projections like needles and pipettes, like the glass pipettes, right. And there are certain other cutting or like a piercing skin the equipments. So hypoderm the especially like the examples are like hypodermic needles and syringes. Those are very commonly used like as a shops. And blades, of course, they use like in nursery or like to tear off any skin of for example to remove some branches of the leaf of in agriculture as well as in animal and human sciences. This is the blade is very common, and it's used like for certain microtomes. Broken lab glasswares, of course, this is also comes under the shops. And of course microscopic glasses and cover slips that we use to make like a certain kind of the slides. Okay, so all these things come under shops. So this is a very important and very sensitive waste, the waste product. So types of waste, there are different types of wastes. For example, the regular waste such as wrapping note papers and pencils, that's commonly also we call it a kind of domestic waste. So of course while we are working in the lab, so we have to produce like this regular waste as well. So because we are working on the lab the notes as well as we are writing something protocols, or we are doing like some the paperwork. Second one is a chemical waste, of course, including all chemicals that we are using in the lab, so fall under like chemical waste. And then is a biological waste. So as I mentioned, in all biolabs, so there are different products that are being used in those particular labs. So for instance, blood samples, blood product in microbiology, the culture plates, and also like animal and fish carcasses, plant material that is infected with the certain the bacteria, fungi, all culture media that we grow, like different strains of the bacterium into the lab, sponges, pipettes, those are contaminated with the biohazards. Of course, then they in the previous syringes and pipettes are different, but we consider as the glassware, but once they are have a contact with the bio products, bio waste, so then it become like a bio waste. Soil, of course, it also contains like some, because in College of Agriculture, most of the soil they are also deal with certain bacteria, soil gloves and containers. So there is a lot of. So yeah, I think while you are working in the lab, so you can understand even there are more low, a lot of other types of biological waste. So once you have understanding about the biological stuff. So, of course, this slide showing the disposal of a chemical waste, and you can see like since as I mentioned we have different types of waste, and according to the type of the waste and according to the category, so we have to dispose of or we have to manage it. For example, aqueous waste, you can see here aqueous waste, and these aqueous waste contain like certain acids, neutral or like the basic chemicals, depending on their pH, and they have less than 40% organic the chemical properties. And the second one is organic waste. For example, those comes like have more than 40% organic chemicals. So they are non-chlorinated or chlorinated, and they are segregated into a chemical that have like a commercial bottles. Okay, so they can also fall like in the organic. Solid waste, of course, there are different types of solid waste, and according to the types of the solids, so we have to dispose of them. For instance, you can see here, so these are the light containers that are used for non-visible or loose, like powders, collect, the unbleached green pails, or like empty into the solid waste drums. Okay, so there are certain like gloves, paper towels, empty like vials, like etc. And of course then solid waste comes like the chemicals. These have they have like a loose powders, so heavily contaminated stuff, it contains. Okay, so there are certain examples. For example, they have a paper, unwanted samples, okay, but those are heavily contaminated. So silica gel, so this is also a very dangerous chemical, of course, disposed in a separate container, so may not be combined with the other types of chemical, the spillage or the storage, the disposals like these we are talking about the above one. So chemicals in commercial bottles, of course, then these come like specialized bottles, and we can also dispose the more like dangerous chemical into these bottles. Then special cases, like shops, of course we cannot dispose of like shops into the normal or any other solid waste, etc. But of course we need a special, I think you visited like a different clinic, so they have like a special kind of storage for the shops, like syringes. Okay, inorganic oxidizing, so place into these particular special separate containers. And of course violently reactive chemicals also, they have like a separate storage. And mercury thermometers, so they contain like a special as well as a separate container. Any uranium, thorium, and of course like a more dangerous stuff called we cannot store, dispose of here, so we need to contact like the special the office. I think most of the technician are like the administration, they know like how to contact those people, and they come and then they can take these disposals. So now like how like different chemicals are being disposed of according to their properties. So if you are working with the different with glassware, so this is really very important to use a proper technique to while inserting these kind of tubes into the beakers. So mostly like they break down the bottom of the beakers. So be careful. So proper techniques you should learn before working on these glasswares. as well as never use these glassware under pressure or vacuum, otherwise they will be a breakdown. And if there are chances, if there is breakdown of because this is even then it's a very normal like routine in the lab. So during washing etc there are breakage of glasses or the tubes and bottles happen, and then the best practice is to dispose of immediately the broken glasses so that it will not hurt not only you but maybe other people also, because sometimes you only the worker only know like there is a breakage of the glassware but nobody like else who is not away, he is not around, but once they visit that particular area might be can get hurt. Okay, so always immediately dispose of these broken glasswares. And the same case is for with the like the solvents and oils, so they should be like segregated. You see like these are the different oils or the salts and the chemicals, so you should be like segregate into correct like a waste bottles. You see there are many different types. So in the last slide we discussed about the different types of the waste, and according to that waste we have to prepare like the containers. So each lab already have they prepared and they mention like the bottles according to their waste. Okay, okay, and of course do not put materials down in the drain. So this is very important, you know, because if we drain certain chemicals, so what will happen? So we will it will affect like our groundwater etc. Okay, so in coming slides maybe we'll discuss this one. So this is not allowed, it is very prohibited, strongly discouraging not to throw any chemical into the drainage. Don't wash any chemical into the drain areas. So this is just kind of the waste segregation like the setups. I think so this is very familiar, mostly like for the normal places you can see like for the paper, metal, glass, plastic, biohazards. Okay, so this is just a one more decent example. This slide shows us a very beautiful the waste management hierarchy, and from this slide we can understand how we can use or reuse or recycle our waste, and then it can be beneficial for our like a society. So for example you can see here, so this is the from bottom to the top, so you can see these are the different colors it's showing, and this is the last resort, and you can say this is the top one or the preferred area. So we will see here, so the lowest one is disposal. So whenever we have certain wastage, so we should go like for disposal. So where we should dispose of, so sometime if there is other facility is not available, then of course landfill happen, like if there is no alternative landfill, dump into that particular, for example go into the mountains and then throw there and then covered with the soil. And the second one is we can dispose of our the waste, and then of course we can recover it, we can get the energy from that, we can recycle some energy from that disposal. Okay, so this is the second the category or the second the state. So then recycle, turning material into new substance. We can recycle, for example there are many waste can be used to make the plastic paper, the plastic waste, etc. So depending on the waste, so if there are agriculture waste can be used for paper or etc. Okay, so recycle. So this is a very good strategy. If we can recycle the waste, then in this way, so it will not like destroy our environment, so it can also be we can make it useful, or you can say reuseful of our disposal of the waste products. So reuse, of course as I mentioned, so there is the certain process if we follow them, and then we can reuse that the waste into the different parts, so that can be reused. So the top or the best one is reduce. So generating less waste is everyday life. So this one is a, so although here is a, we need some certain special maybe technology, so that we will use and produce or reuse or recycle or recover from the waste product. Okay, but everybody at least can this do without any technology. But of course, just if body can participate and morally can support to the society, and then try to reduce the waste generating the materials. So, okay, so for example, try to you know, produce like a less you know, the the waste, whether this is a the paper waste, or any kind of waste, okay, or domestic waste, okay. So this is the preferred area, the preferred portion or the preferred area. So of course, then of course we don't need to spend more energy to reduce or recycle even, okay. So this is of course for lesson for everyone. In last couple of slides, we discuss about the waste, and they have particular specialized or like uh, the specific the container that can use to dispose of that particular waste. But here it's really important to understand, sometimes it's the waste type is the same, but it's non-contaminated, and also it's contaminated. The similar are the same waste, but once sometimes it's contaminated and sometimes it's not contaminated, non-contaminated, okay. And then how, and also sometime like for example the shops or the glassware, they are non-contaminated, then we have to deal separately. So for example, the one waste that is contaminated and the same waste is non-contaminated, so we will not dispose of into the same container. So we need to segregate. For example, non-contaminated waste, we need to segregate into the special or the separate the bin or like the bottle. And then of course if we have the contaminated non-contaminated shops or glassware, so of course we need to separate out like with the into the particular the boxes. So these here you can see like this is the specialized bottle that's uh, these packs come from the company. They can also be used for the broken glasswares. So we can segregate so one for the contaminated as well as one for the non-contaminated. So contaminated laboratory glassware for reuse. Contaminated glasswares which is specifically designed are used for laboratory work, they are discarded in an autoclave bag for sterilization. So this is particularly for the College of Nursing or maybe like the hospital. So even this contaminated glassware, we don't like just put into the garbage and just consider as a garbage. No. So first we have to autoclave it to kill the biological pathogens or the microorganisms. So especially if like the blood samples are contaminated with these glasswares. And secondly, these here you can see these are the different special the containers that are used in the, especially like in the hospital labs as well as like other areas where mostly like the shops are used, and they have contaminated with the special the blood samples, or like the patients those have kind of chronic diseases. Of course these should be autoclave and put into the store into the special like the particular the bags, and then of course dispose of. So this is all about like of course be careful about these because these contaminated the garbage is really important to segregate from the non-contaminated. So in last couple of slides, I give one example, or like I just point out like never throw any chemical into the drainage. So not like throw like any liquid chemical or the waste into the drainage into the lab. So what are, it has certain effects on the environment. For example, hypochlorite, so it will react with organic matter and release into the environment as a treat wastewater, because this is one example. But heavy metals, for example silver, lead, cadmium, so they will also follow this pathway into the water table. So because of course if it's going into the underground, of course then it will leach down and it can become like a part of our water table, and then we can pump out that water, and then mostly like in the particular areas we are going to drink that water. So this is they can cause like of course certain diseases. And similarly certain like water supplies, for example, if they are contaminated with certain hormones, pesticides for example, and other organic molecules into the groundwater, so they can produce a lot of other diseases. So that's why this is really important. While if specifically we are talking about the lab or the laboratory conditions, so don't drain any chemical into the drainage. Okay, so don't throw any chemical into the drain or into the sink. Always dispose of. Okay, although this look like very easy, like because we observe like sometimes some students maybe throw like even the cultures, so that is also like the waste because it contains certain bacteria. So once it goes into the soil and then of course like it's leached down, or it can cause a certain problem, or maybe it can become like the part of our the fresh water. So the once it's pumped out, and then it can cause the different diseases like diarrhea or etc. So this is all about this today's lecture. So very simple two contents, and I hope it's not very difficult. So even if you have any questions, so on each Thursday, so I will be online. Although this is not like uh, restricted for every student to be online, so it's up to you. If you any student have any questions, so I will be there. So thank you so much. Hope to see you on Thursday.